All right, so welcome to the second video of our microbial module. In the second video, we're going to kind of pick up our discussion with the three domains. So we've talked about prokaryotes, the characteristics of prokaryotes, and next we're going to talk about the domain eukarya. And we're going to look at how eukaryotes most likely evolved throughout their evolutionary history. And so just to review, eukaryotic cells are very different from prokaryotic cells in that they're much more complex, they're larger, they have a membrane-bound nucleus that contains the DNA. They also have other membrane-bound organelles like your endoplasmic reticulum, your Golgi apparatus, vesicles, etc. And we believe that the eukaryotic cells evolved approximately 2.1 billion years ago. And remember, this is after prokaryotic cells evolved, and it's pretty long time after. If you remember from the first video, we said that prokaryotic cells were thought to have evolved around 3.5 billion years ago. So we're talking about a billion years, or more than a billion years, in which prokaryotic cells were the only cells that existed on Earth. And we think through evidence like DNA evidence and evidence with, mitochondria and chloroplasts having their own individual DNA, which we'll talk about why that's important in a little bit, that eukaryotic cells evolved from aerobic prokaryotic cells. Okay, and this process happened. So this evolution of eukaryotic cells from aerobic prokaryotic cells, we believe happened through this process of endosymbiosis. Okay. So endosymbiosis is where you basically will have one cell engulf another cell. And scientists hypothesize that during Earth's history, that one prokaryotic cell actually did this. So you had one prokaryotic cell to engulf another cell. And instead of breaking that cell down and using its components for its own cellular structures, the two cells actually live together under a symbiotic relationship. And so that's why we refer to it as endosymbiosis. So endo being part of the endo, where you bring something in from the outside of your cell, so from outside to inside, and then symbiosis with that symbiotic relationship. And so they basically live together and evolved until they become dependent on one another. And so dependent meaning that the prokaryotic cell that was engulfed is thought to have been the precursor to mitochondria. And so now eukaryotic cells can exist without mitochondria because mitochondria is where that cellular respiration happens. And we believe that the genetic material that's in that engulfed cell is similar to archaea. And it also had metabolic organelles that were similar to bacteria. And so basically you have, this cell engulfing what would have been an archaea uh, cell, and basically evolved into the eukaryotic cells that we see today. So basically we have this development of membrane proliferation, so developing of more membranes, and compartmentalization of different processes within the cell. And so basically this endosymbiosis led to the evolution of mitochondria. So the one cell engulfed the free-living aerobic prokaryote. And so aerobic is important because mitochondria undergo cellular respiration, and they need oxygen for that to happen. So it's an aerobic process. Okay. It was shaped like bacteria, surrounded by two membranes. So remember when we talked about mitochondria, we said that it had an outer membrane and an inner membrane. And the mitochondria enfoldings are similar to the outer surface of some bacteria species that we that we've studied. And so, for example, you can see these enfoldings here in these images of bacterial species. Um, and so what can happen is mitochondria can divide on their own, so similar to binary fission, and they have circular DNA. So if you look at a mitochondria today, it has its own circular DNA chromosomes. It has characteristics that are very similar to bacteria and prokaryotic characteristics. And because of that, this is what leads to the idea that an ancient free-living prokaryote engulfed this mitochondria. And while they weren't dependent on one another to begin with, that long period of that symbiotic relationship led to the dependency of mitochondria and on the cell and the cell on mitochondria. So basically the eukaryotic relationship that you see today. And so this also brings up the question, okay, well, what about plants? They have chloroplasts. So how did these plant cells evolve? So if we actually look at chloroplasts and the structures of these chloroplasts, we see that chloroplast is an organelle found in plants. It's important for photosynthesis. It has starches, fats, proteins, pigments, but it also has two or more membranes. It has those double membrane structures. It also has circular DNA. Chloroplasts also divide in a function that's similar to binary fission. And they're very similar in size and shape with photosynthetic cyanobacterium that we said were we think were the first photosynthetic organisms to evolve. And so if you look at the structure of a chloroplast and compare it to a cyanobacterium, very, very similar, right? Very similar structures. And so what scientists think is based off of the similarities in structures, based off of the circular DNA, and they also did DNA work with mitochondria and chloroplasts, and comparing those with bacterial cells and prokaryotic cells, that another process or another episode of endosymbiosis occurred where a cell engulfed a photosynthetic cyanobacterium and they became dependent upon one another. And so basically we have two episodes of endosymbiosis. You have a primary endosymbiosis, and then you have a secondary endosymbiosis. And we think it's secondary because remember, plant cells actually have chloroplasts and mitochondria, okay? And so the way we think this happened is you have this heterotrophic cell, right? And initially it was a cell that did not have a mitochondria, right? It engulfed a prokaryote and it led to the development of that mitochondria we see here. So now you have a heterotrophic eukaryote cell, okay? Then you have this cell to engulf a cyanobacterium, okay? And after that happens, one of the membranes surrounding that cyanobacterium is lost. And then you end up with a second endosymbiosis, right? Where you have the prokaryote, not prokaryote, the cyanobacterium being engulfed in that heterotrophic eukaryote, leading to a vestigial nucleus and a plastid, right, which is what we see with the chloroplast today. And so we think that this happened after mitochondria were present and mitochondria had evolved. So you have that initial endosymbiosis with the mitochondria, and then you have another process of endosymbiosis, another episode where the cyanobacteria were engulfed. And only your terrestrial plants and algae have chloroplasts, but all have mitochondria. Okay, and so that's an important distinction that leads to this hypothesis here. So for example, your red and green algae share very similar DNA sequences with photosynthetic cyanobacteria, meaning that they are very similar in that they share a common ancestor, right? And that's one of the reasons we believe that this endosymbiosis process happened throughout the evolutionary history of eukaryotic cells. So here we have another image showing that endosymbiotic theory. So you have a proto-eukaryote, right? It has these membranes being developed and formed. They engulf an aerobic bacteria that leads to mitochondria. Okay? After this modern heterotrophic eukaryote is formed, you get a second endosymbiotic episode where they bring in a photosynthetic bacterium, and that is now incorporated into the cell. And so now you have a modern photosynthetic eukaryote. This organism can photosynthesize. This organism is a heterotrophic organism that has to consume molecules in order to put into the mitochondria to develop energy. So these two endosymbiosis episodes led to the development of heterotrophic eukaryotes and photosynthetic eukaryotes. Okay? And so if we look at the domain of eukaryotes in general, so overall, so after this has happened and evolution has happened and led to all of the diversity in eukaryotes, we end up with 4 kingdoms that exist in the domain eukarya. So remember, we have domains, kingdoms, phylums, etc. Right? So in the domain eukarya, there are 4 kingdoms: fungi, plantae, animalia, and then protista. Your protista is your protists, fungi is your fungal species, then your plants, and then your animals. Fungi, plants, and animalia are well-defined monophyletic groups. Monophyletic groups meaning we can trace all of the descendants back to a common ancestor. So they all form this relationship, this grouping that contains the ancestor of animals and all of its descendants, ancestor of plants and all of their descendants, etc. However, protist is not a monophyletic group. They are actually, we think, probably made up of several kind of other kingdoms, and this group has not been kind of sorted out yet. You can think of the kingdom protist as the eukaryotic junk drawer. So basically, if it's not a fungi, a plant, or an animal, it goes into the protist kingdom, and it's basically left to be sorted out later. It's basically what you can think of the protist kingdom for eukaryotes. A lot of work still needs to be done in all of the groups, but especially for protists, because it doesn't form a monophyletic clade, and the relationships, evolutionary relationships, are really in poor shape. So here we're looking at the kingdom protist with some examples. So you have a single-celled organism here. You have two single-celled organisms, and then kelp is actually a protist species. So kelp isn't a plant. So like the kelp forest off the coast of California isn't a plant. It's actually a protist. So it's an algae. It's a species of algae, some multicellular protists, whereas these are single cellular protists. If we look at the evolutionary relationships of these groups, you can see that protists are kind of all over the place. You have some protists that are closely related to plants. You have some protists that, are closely related to each other, but they all don't form a monophyletic group. Whereas plants, you have this one ancestor that leads to all of plants. Animals, you have this one ancestor that leads to all of animals, one ancestor that leads to all of fungi. So these are monophyletic groups here. Whereas protists, again, it's just kind of a mess trying to figure that out. And so again, it's anything that's not an animal, a plant, or a fungi that is a eukaryotic organism. Usually you find protists in generally aquatic environments or aquatic-based environments. And then also some parasites that infect plants and animals also fall into protist species. Now, protists can live a variety of different ways. It can be solitary, they can be colonial, they can be parasitic, they can be in symbiotic relationships, they can be heterotrophic, they can be autotrophic, or you can classify them as both or omnivorous. Photosynthetic protists we often refer to as phytoplankton. So that's the phytoplankton that's floating kind of in aquatic environments that photosynthesize and kind of lead to what you think of like your algal mats or those types of structures you see in aquatic areas. Heterotrophic protists are known as zooplankton. So these actually eat your phytoplankton or your diatoms or some of your other smaller floating organisms that are in these aquatic environments. Protists can reproduce by several different methods, asexual and sexual. Sexual methods are very diverse and actually depends on the group, so we're not really going to kind of go into that. For asexual reproduction, it could be binary, so 2 daughter cells, or they can produce multiple daughter cells through that binary fission. They can also bud off, so you can have a piece of that organism break off and grow into an entirely new individual, so through budding. And so very, very diverse in terms of reproduction. And what's also interesting is that they can even switch between asexual and sexual modes of reproduction. So very interesting group, very complex group of organisms, which is also what makes it so difficult to classify these individuals into this kind of organized relationship. So on the left, we have Paramecium. In the middle, we have amoebas. On the right, we have pretty common photosynthetic organism called Euglena. Paramecium will kind of move around. They have these cilia, and they actually are heterotrophic. They'll engulf other organisms. Amoeba is kind of this, organism that you can kind of think of it like using these little pseudopodia appendages to kind of move around and kind of slink across the ground, and it uses the pseudopodia to actually attach to solid surfaces and basically pull itself forward. And then Euglena has a flagellum that it actually uses to propel itself throughout the water column. Now, we said the way that they can get food is a variety of different ways. So autotrophic, heterotrophic, and both. Whenever we look at, say, for example, autotrophic organisms, they are you could be photoautotrophs, so photosynthetic, have chloroplasts. They can be aerobic or anaerobic, so they can use oxygen or not use oxygen. And then they also can get food through heterotrophic ways, which are known as phagocytosis. So basically a form of like endocytosis, where they ingest food particles from the outside of their environment, they bring it in, and an enzyme and a lysosome will break that down and allow that material to be incorporated into their cellular structures and allow the energy to be used in respiration, etc. And so this is an example of an amoeba, amoeboid organism. You can see the pseudopodia, the nuclei, and you can see the chloroplasts that are used for the photosynthesis of that organism. Here we have a paramecium, which is a heterotrophic organism, uses the cilia to kind of float around and kind of move back and forth. It has contractile vacuoles that allow it to control the water volume, so excrete excess water. And it also has an oral groove, which is basically its mouth. And so it brings in food and it excretes waste out of that oral groove. So very cool, interesting organisms here. Okay. And so the domain Eukarya is actually broken up into 6 supergroups. Each of these supergroups are monophyletic, and so what that means is that they all come from a common ancestor, like I explained earlier. These supergroups are developed based off of DNA sequences, so genetic sequencing data, and protists actually exist in each of these supergroups. So protist in general is not a monophyletic group because you find them in each of these groupings. However, plants and animals and fungi are only in kind of one of these groupings that lead to monophyletic relationships. So for example, animals is here, right? Plants are here. And then your fungi, I'm blind, I can't, are down here, right? And so those are monophyletic groupings of your plants and fungi and animals. Whereas all of these other kind of taxa or groups that are part of these super groups are prokaryotes, right? So like your so these are not prokaryotes. Your fungi's not. I can't remember what the nuclearids is, but for example, slime molds is here. That's a prokaryote. Red algae are prokaryotes, and they're not closely related. Right? Red algae is actually more closely related to land plants than slime molds, and so that difference doesn't make them monophyletic. So now looking at some pathogens and diseases that come from prokaryotes, not prokaryotes, but protists. I said prokaryotes earlier too, but I meant protists. Sorry about that. Your human pathogens, like your plasmodium. So for example, malaria, it comes from this Plasmodium falciparum species, and this plasmodium is transferred through a mosquito, and so it actually destroys your red blood cells and can cause severe anemia and fever episodes that can be fatal for the organisms. And so here's actually how that plasmodium is transferred. Very complex life cycle. You don't have to remember this, but for example, you have the protist organism to let's find the mosquito. So it gets in the mosquito, right? It goes through a mitosis form in the mosquito, and the mosquito will then inject the plasmodium into a human. That plasmodium will then go into the liver and undergo some stages where it will multiply, and then it enters the blood, and it affects the blood, and then it will have gametes that are produced again that can be picked up by a mosquito that feeds on the blood and then undergoes that life cycle again. And the plasmodium has to go through this specific life cycle in order to reproduce and function. So if you remove any point of this life cycle, you will break the reproduction of the mosquitoes and of the plasmodium. Now, another example of protist pathogen is African sleeping sickness, um, which is the tsetse fly vector. And so the trypanosomes, which is the species of the protist, is actually brought about from the fly to the human, and it actually affects the nervous system. You can see the protist in the blood of the sample here. And if you don't treat it, it is fatal, and will lead to death. You also have another species that is damaging to the heart and the digestive system, and again fatal if not treated. And so these are, basically parasitic protists that are transferred to humans through specific vectors. Again, it could be mosquitoes, it could be tsetse flies, or it could be a specific insect or another bug. You also have some other infections that are due to protists. So these downy and powdery mildews that are, for example, found on grape plants, potato. So from potato blight is all based off of protists. So all of these diseases and plant parasites are protist based. Now, just like with the prokaryotes, there are bad prokaryotes, good prokaryotes, and there are bad protists and beneficial protists. So, for example, food sources can come from protists, mutualistic relationships. So, photosynthetic producers for some species. So, for example, protists can live in a symbiotic relationship with organisms and develop food for them, and that other organism can provide a place for that protist to live. Or the protist can serve as a digestive enzyme to help with the breakdown of food so that the organism that it's living in can incorporate that organic material into its body or structures. And so, for example, polyps have a symbiotic relationship with dinoflagellates, which are the protists, and the dinoflagellates help the coral polyps obtain food, and the coral polyps provide a place for the dinoflagellates to live. Protists are also important for decomposers. So you have a fungus-like protist that will break down nitrogen and carbon and return inorganic nutrients to the soil and water, and again are very important for those nutrient cycling effects that we talked about with the prokaryotes themselves. And so that's it for the eukaryotic portion of this module. Next, we will finish up with the fungi kingdom in our last video for this week's module. Yeah, so we'll see you there.